Hello, grade 12 learners of South Africa. Welcome to another in the episodes of Mindsets Life Science for grade 12s. I would hope it was for grade 12s because why else would you be watching? Well, last time we did the whole male and female reproductive system. You learned about the basics of gametogenesis. We also did the hormones. In males, we have testosterone, and in females, we have estrogen and progesterone. So now we're going to go on to what the details are of gametogenesis and also hormone control and how those hormones in our bodies control everything that happens. Now, in grade 10 and 11, you would have done proteins and you would have done why proteins are essential in our bodies. It's because all hormones, except the sex hormones, are made of proteins. So, except the sex hormones, we are now going to focus on those sex hormones that are made of fat. And that is why when people become severely thin or there is severe malnutrition and they lose a lot of body weight and body mass, and the body literally starts to eat their own cells, eat in inverted commas, you start to metabolize and break down your proteins and convert it into glucose and urea so that you can survive. That's why people get so incredibly thin. They forget or lose their periods, especially in females. And in males, the sperm production just drops right down because it's the one process the body can sacrifice in order to keep you alive. All right, so here we go. Reproduction, we're doing gametogenesis and hormone control. Now, people, if we look, there are three processes in reproduction. The first process is gametogenesis. Why? We have to produce those little gametes. Remember, gametes are haploid. And the fact that they are haploid, they are going to have half the chromosomal content of a diploid cell. So when we take a haploid gamete and a haploid gamete and we fuse them together, we then have a diploid zygote. So a haploid gamete from the male, a haploid gamete from the female, we fuse together and we have procreation. We then create a new organism. So first part has to be gametogenesis. Then we look at fertilization. Clearly, that is the fusion of the gametes. So here we're going to have the haploid plus the haploid is going to give us diploid, which is the diploid zygote. And then we have the embryo development. And here, mitosis is important. But to form the gametes in gametogenesis, we are going to have meiosis. And if you recall, when we did meiosis, I said to you, meiosis is for sexual reproduction. So that is where we're going to have reduction division to convert the diploid cells into haploid cells so that sexual reproduction can occur. Mitosis, remember the functions and the roles of mitosis was for growth, for repair, for replacement of cells, and then also for reproduction in unicellular organisms. They only have one cell. So that one cell has to do everything, including reproduce. Okay, so we're going to then look at the embryo development. So it starts off with gametogenesis, producing those little gametes. Then fertilization is the fusion of those gametes. And once they have fused, we now have the diploid zygote, which then develops into an embryo. All right, now, gametogenesis, we are going to discuss in diagrammatic form what happens in meiosis. So the specific meiosis that occurs within the male and female reproductive systems, and we're doing human reproduction here, but a very similar process occurs in the anthers of a flower and the ovary of a flower, because remember, you're also going to have meiosis occurring there to produce the egg cells and the male gametes in the anther, which then we have as pollen. All right, now, this is the production of haploid, boys and girls, haploid gametes in the germinal epithelium. Now, germinal means where growth occurs. So where the process actually occurs, it will be germinal. Think of when, in past years, when we spoke about germination of a seed. Germ means to grow, to make new. So germinal epithelium, very important, you must know this, germinal epithelium by a process of, we've done this, meiosis. Now, when gametogenesis takes place to produce the sperm cells, Now, sperm cells, we call it spermatogenesis. And when it is the female to produce little egg cells, we call it oogenesis. Now, just think of an O is the same shape as an egg. So it's to produce the egg cell. So gametogenesis in males is called spermatogenesis. And in females, oogenesis, because you're going to produce those little eggs. Okay, so please remember spermatogenesis and and oogenesis. Now, what I've done is I've put together for you a diagram which represents the male reproductive system, so spermatogenesis and oogenesis on the same diagram. Now remember, they are both processes that are in fact gametogenesis, all right? Gametogenesis in males is spermatogenesis and in females is oogenesis. And I hope that has now gone into your heads and you now know it off by heart. All right, if we look at this, we start off with the germinal epithelium. That's very important. The germinal epithelium cell is where creation is going to occur. So in the male, we call it the spermatogonium, and in the female, the oogonium. So that's the special name for the germinal epithelium, where these new little cells are going to be created. We start off with the germinal epithelial cell. In the male, it will be the spermatogonium, and in the female, the oogonium. Mitosis and cell division occurs, and we have repeated division so that we now have many cells. Okay? These four little structures here, which started off with this one structure that has now undergone mitotic division, divided by mitosis, so there are many. Okay? We call them the spermatogonia, and in the female it is oogonia. This is why it is easier to learn this diagram together, so that you're doing spermatogenesis and oogenesis at the same time. And then you have both. I would actually copy this. Now it's in your X sheets. So as soon as you get your X sheet, take this, cut it out and stick it up on that fridge or behind the toilet door so that you can look at it again and again and again. And after a week, you will know the processes. So we start off with the germinal epithelium, which is the spermatogonium, or the oogonium if it is in the female. That undergoes mitosis. That's step one. We have mitosis, which then forms many cells, many spermatogonia, if it is happening in the testes of the male, or if it is happening in the ovary of the female. You're going to have your oogonia or your oogonia. Those oogonia then develop into the primary spermatocyte, and look, people, it is still diploid. And in the female, the primary oocyte, which is also still diploid. No meiosis has occurred here. Have a look here. No meiosis. All we've had is mitosis, okay, to form the spermatogonia. The spermatogonia then grow. We have growth taking place, and that growth is to increase the size of those cells. And they develop the spermatogonia, mitosis. Spermatogonia is step 2. They will develop into primary spermatocyte, and it develops here into the primary oocyte. But they are still diploid. No meiosis has occurred, all right? Now watch this. You'll see there's a line that's been drawn all across here. That's where meiosis is going to take place. Now, we have meiosis 1. Let's use our magic pen. Meiosis 1 and cell division. Now, we know that during meiosis 1, that is when we have genetic variation occurring. Why? Because we have crossing over. And when that crossing over process occurs, you have a piece from the paternal chromosome and a piece from the maternal chromosome, and they cross over and break off and reattach to the other chromosome. So meiosis 1 is incredibly important, especially prophase, because that is what results in genetic variation. And people, this links in with diversity, which is module 4 of your syllabus. So this is what causes diversity. This is what makes us different. And it's also what makes you a combination of your mom and your dad's grandfather and his grandmother and his great-grandfather and his great-grandmother, and the same on your mother's side. So that you are not just a clone, you have genetic variation within you. All right, so meiosis 1, we have cell division occurs. All right, now we go to, after meiosis 1, which is step 4 of the primary oocyte and the primary spermatocyte. A site means a cell, so that cell is going to change now. Meiosis occurs, and that will result in a secondary spermatocyte. But now look at this. Your primary spermatocyte is diploid. Your secondary spermatocyte is going to be haploid. Why? Meiosis 1 has occurred. So after meiosis 1 in the female, that primary spermatocyte is reduced to the first polar body, which is going to provide nutrition for this little egg cell, and the secondary, it should be secondary, people, secondary oocyte, and that is going to be haploid. Now this is what's going to develop into the egg cell. Then look at this, we have meiosis 2 and cell division occurs. Now we're sitting with the reduction division resulting in a secondary spermatocyte, which is haploid, or a secondary oocyte, which is haploid. What happens now? Our fifth process is going to be meiosis 2. So meiosis 2 has occurred, and we work from there. All right, here, spermatotids, what are we going to have? The spermatotids are haploid. So meiosis 2 is going to result in duplicating our secondary spermatocytes, which are haploid, into 4. What we would have is here we would have, for example, XY, going back to the genetics we did. So this one would be X, that one would be Y. So we would have 2 X sperm cells and 2 Y chromosome sperm cells. And what are the spermatotids when they mature? They mature, and they are now going to form our 6th step, which is our sperm cells, which are haploid. And in the female, this polar body is going to now split into 2 polar bodies. So we have the first polar body, the second polar body, and the third polar body. They are all haploid, and they are going to form nutrition for this little egg cell, which is still only going to be haploid. All right, so let's just go over this again. They could give you this schematic representation in an exam. But you need to know this because at the end of the day, your whole process of spermatogenesis or oogenesis, you can either get one half of this diagram, or you can get the whole diagram, or you can get this diagram represented as either the male or the female body. So you need to understand it. So let's just go through our steps again. All right, and I know I'm laboring the point, and if you've got it and you remember it, great. Then check yourself now as I go through it. Maybe close your eyes and listen. For those of you that don't have it, well, listen carefully because we're going to go through it a second time. We start off with the germinal epithelium. In the male, we have the spermatogonium, which is the germinal epithelial cells. First step, we are going to have mitosis. What mitosis does is it now results in 4 identical cells, which are called spermatogonia. And this happens in the testes. The spermatogonia are going to grow and develop into primary spermatocytes, which will be diploid. The diploid spermatocyte undergoes meiosis and develops into the secondary spermatocytes, which are haploid, people. Very important, they are haploid. Then the next phase is going to be meiosis 2, where those spermatocytes are going to undergo secondary meiosis or cell division and form spermatotids, which are haploid. The spermatotids are going to mature, and as they mature, they are going to mature into 4 little sperm cells that are haploid. In the female, we have our germinal epithelium called the oogonium, and it undergoes mitosis. And you know, mitosis has double division, so we end up with 4 identical daughter cells occurring in the ovary. Those daughter cells, called the primary oocyte, is going to undergo growth. It is still diploid. All right? Now, we have meiosis occurs. That primary oocyte now develops into a secondary oocyte, which is haploid, because of meiosis and cell division, and 1 polar body. Now, duplication of the polar body results into a first, a second, and a third polar body, which is all haploid, and the egg cell, which is haploid. All right? And that is gametogenesis. In males, it's called spermatogenesis, and in females, it is called oogenesis. I say it that way so you can spell it. It's oogenesis. Or you just say oogenesis, which is fine. All right, now we get to spermatogenesis. It's just simply what I have explained to you in written form. So this is going to be quick. We go with, at puberty, the male will reproduce about 120 million sperm per day. Is that not amazing? Each male that is now sexually awakened and mature is going to produce, wait for it, 120 million sperm a day. Now that is an astronomical amount. Why? Because we need to make sure as organisms, as living organisms, that reproduction can occur. And the egg cell sits there. The egg cell's protected inside the female's body. The same with a plant. It sits in the ovary and it's protected. It's the poor little sperm cells and the male gametes. There have to be plenty to make sure that one of them gets there. Think of pollen. Think of how we all sneeze and wheeze and get hay fever during spring. Because there is so much pollen around. Why? Because nature is ensuring that the female can be fertilized. Because if the female is not fertilized, guess what? No procreation occurs. So 120 million sperm cells. Then the germinal epithelium produces spermatogonia, which is what we've said, and the spermatogonia develop into primary spermatocytes. Okay, then the first meiotic division, and remember, it results in a haploid secondary spermatocyte. Second meiotic division results in the spermatids. Now, very important, the cells of Sertoli. That sounds so wonderful. It sounds very grand. What do? They actually provide nutrition or food for the little developing spermatids. And only when those spermatids are now ready and grown up and ready to go, then they are classified as sperm. So our little spermatids, they get fed by the cells of Sertoli. Please, people, look at the spelling, S-E-R-T-O-L-I, Sertoli. And then the spermatotids, as soon as they've sort of matured a little bit, the cells of Sertoli have provided them with a bit of nutrition so they can just get ready because they have to swim hard. So they can get ready for the big swim. And what do then? They move into the seminiferous tubules. Now, these words are quite difficult to remember, so look at the spelling. Seminiferous, because it's part of semen, and semen is your sperm cells plus all the liquids that help the sperm to swim. So your seminiferous tubules to mature. So what is the function of the seminiferous tubules? It is where the spermatids mature. And when they have matured and they are now ready, it's almost like a training camp for Olympic swimmers. So your spermatids are now sitting and they are training, boy, and they're getting ready for the big swim, for that competition to get to the egg cell first. All right, and where do they do this? In the seminiferous tubules. Function of the seminiferous tubules. Now, if you look at your egg sheets, what we've done there is we have taken all the different structures within the male reproductive system in a table form, and then you have all the functions. You must know, people, you've got to know the structure versus the function. All right. Here we have spermatogenesis and exactly where it takes place in the actual male reproductive system. Here we have the testicles, all right? And in those testicles, we have millions of seminiferous tubules. It's meters and meters of seminiferous tubules, all right? And this is where our little spermatids are now going to mature, and they're going to get ready. Now, what we've done here, they're going to move from the seminiferous tubules. They move in the vas efferentia. From the vas efferentia, they move into the epididymis. And then from the epididymis, they are going to move out via the vas deferens out through the urethra. All right? Now, you know all of this from the male reproductive system. Okay, for the first Saturday extra 8 that we did. Now that here, we're concentrating on what happens inside these seminiferous tubules, all right? What is going to be happening there? This here is a little cross-section. If we take that tube, that seminiferous tubule, and we cut it, and we then put it so that we can look at what is in that slice, this is what we are going to see. All right? Now, we're going to have the basement membrane, because there's got to be a basic membrane down at the bottom on which everything happens. So we have this basement membrane of the seminiferous tubule. Then we have all the different processes that occur. So first thing we're going to have here is our spermatogonium undergoes mitosis. So once it's undergone mitosis, it now becomes the primary spermatocyte. So let's do it like this. This is 1, and then it goes from 1, it goes to 2, okay? And from 2, it then develops into the secondary spermatocyte. So this would then be 3. From the secondary spermatocyte, okay, we are now going to have the early spermatids. There are little sperms that are formed. And why are the cells of Sertoli important? Because they are going to find this, by the way, should go all the way through to there. Your hang on, let me just get this to be pen and not disappear. Your cells of Sertoli, and their job to feed those little sperm cells. So, and look what we've got here. There are early spermatids, and they are now going to develop into the little sperm cells. Let's call them sperm cells because they actually are sperm cells or male gametes. So our little sperm cells have now, that's how they are going to develop, and they're all going to be in different processes within the seminiferous tubules from the germinal epithelium. All right? Okay. Now the sperm cell. And this little sperm cell is very, very, very special. Remember, when we did genetics and we spoke about the production of the male and the female gametes and how they pass down and what the chances are of having a male or a female baby, it was determined by the male. Why? Because the male had an X and a Y chromosome, whereas females only have X or X. So X or X, well, take your choice. It's got to be X. But if it's X or Y, it's the male that determines the sex of the baby. So those sperm cells are really very special because they have to swim like anything to get to the egg cell. And the little egg cell sits there and it decides, do I feel like letting this whole bunch of millions of sperms that are now just attacking my outer wall, do I let them in or don't I? I like that one. In you come, hun. And it only allows one little sperm cell in. So the sperm cells have to really be good. So here we go. The sperm cell is about, now look at this, 2,5 micrometers. That is tiny, 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 tiny in diameter and about 50 micrometers long. So they are very, very, very small. The sperm cell structure, what has it got? It's got a head, and inside the head we have a haploid nucleus. It has a short little neck, and then it's got this long tail to help it to swim to the egg cell. What does it swim in? It swims in that seminal fluid, and which is made up of a whole lot of secretions from the different glands, which we're going to get to. All right, here we have our little sperm cell, and it is ever so cute. Look at it. It's got its little head, and it's got the acrosome, which is a sort of a lining across the top, and there's the little head, and there's the tiny little neck, and then it's got like a sort of a middle section, and in that middle section, we have many mitochondria. Now, people, cast your minds back. In grade 10, you did cells, and you did all the organelles within a cell. What was the function of a mitochondria? Come on, think. It was to provide energy, because your mitochondria take oxygen, it takes glucose, and it combines those two to give energy. So wherever you are going to need anything in your body, where energy has to be provided, you are going to have lots and lots of mitochondria. So many mitochondria. Why? For ATP. Because what is ATP? ATP is adenosine triphosphate, but you only need to know ATP, and it is energy. So centriole, and then the axial filament, and what do we have for the rest of it? There's our little sperm cell with its very long tail. Why? Because this tail assists with motility. Motility means to be mobile, to move, to swim as quickly as possible, and the little tail just goes chick, 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 and it swims. All right, now what we've done here is we've taken a cross section, so we've cut through it, and we're now looking at it this way, round. So just think of a carrot. If we take a carrot and we cut into the carrot and we look at the carrot this way, this is what you are going to see. You're going to see little microtubules, and then in the middle, you're going to have the central microtubules. So the energy for this swimming, for the motility and the movement of the tail, is going to be coming from the ATP of the mitochondria. So little sperm cells, very special. Now, if we look at the hormonal control of spermatogenesis, and this really is quite important. We're now going to say, right, how does it work? Nothing in your body, and please understand this, nothing in your body, there's not one process that works without two things. One is enzymes. Why? And you did this last year. Because enzymes lower the activation energy and speed up the reaction. Our bodies could not function if we didn't have enzymes assisting us with all our reactions, because we would literally lie in one place, be completely sedentary, and just have tubes feeding us so that we had enough energy for our processes to occur in our body. Now, think about it. I'm standing here and I'm talking to you. My arms are, because I can't speak without my hands, but my hands are going, my brain is going, my mouth and my eyes are going. Everything is moving and processing. At the same time, my digestive system's digesting my breakfast, and my kidneys are doing their excretory process, and my brain is working, and my cardiovascular system's working. People, all those things require energy. All right, so we need our enzymes because they lower the activation energy for every single reaction in our bodies, and you must know this. They're going to expect you to know this in matric. All right, and then we think of hormones. Now, what do hormones do? Well, every organ, every cell, every tissue, every bit in your body needs to be told to do something. Think of your body and the cells in your body like they're like a 5-year-old child that's about to have bath time. That 5-year-old child's not going to get into the bath to have bath time unless they are told to do so, all right? And then you got to get the kid into the bath. And then they bath and they mess and they draw on the walls with soap, chalk, and whatever the case is, and they play with their toys. And when they finish, they get out of the bath and the whole floor is wet. Everything's a mess. That 5-year-old child will not tidy the bathroom up and hang the towels up after they've bathed unless you tell them to do it, or else someone comes along and does it for them. And that is how anabolic steroids work. They come and they do it for your body. So your body gets lazy and things shut down, and that's why people that take anabolic steroids end up with very bad skin, and they end up with all kinds of hormonal issues because you've got someone else coming in and doing it for you. But your hormones are like your mom. And they turn around and they say, listen, hun, clean this bathroom up, pack your toys into that basket, let the bath water out, hang your towels up, and make sure the floor is dry. Your mother would then, in that case, be the hormone. All right, so hormones are incredibly important because they make sure that processes occur. So let's look at hormonal control, because those sperms are not going to be produced unless the hormones tell the testes to produce sperm cells. And the same we are going to go through when we do the female. All right, so hormonal control of spermatogenesis, incredibly important. How? We have a structure in our brain called the hypothalamus. And the hypothalamus is the ruler, the state president, the CEO of a company. In your brain is the hypothalamus. It regulates your body temperature. It regulates your need for sleep. It regulates your sex drive. It regulates your heartbeat rate and your breathing rate. It regulates your metabolism. Without the hypothalamus, nothing in your body is going to work. And the hypothalamus controls all your hormone systems in your body, called the endocrine system. So the hypothalamus is the CEO, and the hypothalamus turns around and it releases a hormone called gonadotrophin releasing hormone. Now, thank heavens, all you need to know is GnRH, and it's a little n. So gonadotrophin releasing hormone. And that gonadotrophin releasing hormone then stimulates the pituitary gland, which also is in the brain. Now, if I took a knitting needle and I stuck it here above your ears, and you put that knitting needle through there, and you go to the center of your head and you put a knitting needle through there, where the two knitting needles will meet, that is where your pituitary gland is. And just above it is your hypothalamus. So the pituitary gland, and it's called the master gland because it has direct link with the hypothalamus. So the hypothalamus tells the pituitary gland, listen, please can you? And then the pituitary gland does, and the pituitary gland then tells all the other organs or glands that release hormones to do their job. So you could almost think of the hypothalamus as the CEO of a company, and your pituitary gland is like your financial director, because without money a company can't run. So the financial director will say, well, marketing director, you can do this, and sales director, you can do this, and everybody else can then do what they need to do. All right, so hypothalamus then releases gonadotrophic releasing hormone, and that stimulates the pituitary gland to release two hormones. And those hormones together are called gonadotrophins. And I've left the O out here. So gonadotrophins. And the gonadotrophins, remember, the testes are called gonads. They're sex glands. So they are the gonads. So gonadotrophins. Now, what are these two hormones or these two gonadotrophins? They are follicle stimulating hormone, and you need to know FSH. Now, it stimulates the follicle, so it's really not difficult to remember. Follicle stimulating hormone is going to stimulate spermatogenesis to occur. Luteinizing hormone, or LH, stimulates the synthesis of the hormone testosterone. So, follicle stimulating hormone is going to make sure that spermatogenesis occurs, and your luteinizing hormone is going to make sure that there's testosterone. All right? And what does the testosterone do? It's produced by the Leydig cells in the testes, and that is for all your secondary sexual characteristics that males have. Let's recap. Hypothalamus releases GnRH. The GnRH stimulates the pituitary gland to release gonadotrophins. The gonadotrophins are FSH and LH. Your FSH stimulates spermatogenesis, and your LH is going to stimulate the synthesis of testosterone by the Leydig cells. So what are the Leydig cells' function? To produce testosterone. How do they produce testosterone? Because the pituitary gland releases luteinizing hormone, which tells the Leydig cells to do so. Okay. Now, let's look at these questions. Question 1 is taken from the National Senior Certificate Preparation Examination for 2008. All right, now the diagram below represents a cross-section of a human seminiferous tubule. Now there are no seminiferous tubules in females, so immediately you know this question is going to be about the male reproductive system, in which, look here, spermatogenesis, in other words, the production of sperm, is occurring. Study the diagram and answer the questions that follow. So, people, what do we have here? They give you a lovely diagram and they say to you, we have the connective tissue between the seminiferous tubules. There are the cells of Leydig, and what is their function? Come on, to produce testosterone. Then we have the spermatogonium, and the spermatogonium are still diploid, remember, and they undergo meiosis, at least mitosis and then meiosis, and we have the spermatotids. And what is B? We have to always put our labels in. Those are going to be our little sperm cells, all right? So, and this is the portion of the seminiferous wall. So we've done that already, so let's look. And it tells you, your heading, it's the cross-section of a part of the human seminiferous tubule showing spermatogenesis. So let's look at our questions. Name the hormone produced by the cells of Leydig. Come on, we've just done this. It's short-term memory. It is testosterone. All right, that's 1 mark done. Name one function of the hormone named in question 1.1. People, you've got your secondary sexual characteristics. So you would have to name them, which would be a deeper voice. Just think of guys when they start to mature, they get a deeper voice. They have pubic hair. They have facial hair. Those are all secondary sexual characteristics, all right? That you could say, you could have given that answer. You could also say that you have rapid or quick increased physical growth. I wonder if you know this. Between the age of 0 and 5, you do more physical growing than you do in your entire life, by the way. All right. And a child of 24 months, has reached half its height as an adult. There's a variation of maybe 1 or 2 centimeters. But if you measure a child at 24 months and you double it, quite literally, that is almost the length that they're going to be. So they could be a centimeter or 2 taller or a centimeter or 2 shorter. But that gives you their height. But by the time a child is 5, they've done pretty much 50% of the learning that they will do for the rest of their lives. Okay, that's number 1. Number 2, if you think of growth, when do boys grow the most? They turn into men from about the age of 14, 15. So grade 7 and grade 8, grade 8 and grade 9 around about there. By grade 10, they are now getting bigger. And by 11, that's it. They've now gone through puberty and they are now tall and they need to just fill out a little bit. So that is also because of testosterone. And the third importance or third function that you could give is the maturation, or maturing is easier to spell, the maturing of the reproductive organs. Now I'm just writing rep, you need to write this out in an exam, and also the production of sperm. Okay, or sperm cells. That is what this testosterone causes. Now, how many chromosomes are in each of the following? Now, people, here they expect you to know the process of spermatogenesis. The spermatogonium cell, now remember the spermatogonium cell is while mitosis has occurred but not meiosis. So this would be diploid. It would still be diploid. Whereas your spermatotid has undergone meiosis, therefore it would be haploid. And it's your spermatid that is now going to mature and become a little sperm cell because it's an immature sperm cell. Explain the importance of spermatogenesis. Let's just get ourselves space here. Explain the importance of spermatogenesis as a mechanism to introduce variation, and refer to prophase 1 and metaphase. So what are we going to say here? We're going to say, first point, homologous chromosomes. Remember the homologous chromosomes in meiosis? Something you need to note in this question is they're combining reproduction, spermatogenesis, and meiosis in one question. So you've got your homologous chromosomes, which are in the primary spermatocytes. Okay, and they exchange segments of the paternal and maternal chromosomes during crossing over in prophase 1. Oh, yeah, what am I doing here? Let's just fix that. Okay, now with that exchange, variation of traits results. Or you could simply say that with that swapping over, with that crossover, traits from the mother and the mother's chromosome or the maternal chromosome and the paternal chromosome are exchanged and swapped. All right, so we'll put a little bullet point there. Our next little bullet point, that, because remember it's 4 marks. We've now spoken about prophase 1. Now we go to metaphase 1 and we say during metaphase, and it's not just any metaphase, it's metaphase 1. All right, the homologous chromosomes what do? They line up at the equator. Because remember, metaphase is the middle phase. They line up at the equator and separate in, and this is what's important, independently. That's your operative word, the most important one. Independently during karyokinesis. And that also results in variation. Now, if you recall, when we did meiosis, I said to you that and genetics, every single time reproduction occurs and gametes are produced, we have 4 chances of a characteristic being transferred. 4. And they are 4 chances every single time. That is why we also said we discussed this when we did the determination of sex during genetics. We said that every single time reproduction occurs and a new baby is created, the sex of that baby, there's a 50% chance of it being female and a 50% chance of it being male. Let's say a couple had 8 children. All 8 could be boys or all 8 could be girls, because every single time they have fertilization occurring, the combination of gametes, there are 4 chances, 50% that it'll be boys and 50% that it'll be girls. And that's what we're working on here. All right, people, so we've covered metaphase 1, we've covered prophase 1. Prophase 1 is the crossing over and the swapping of paternal and maternal pieces of chromosomes. And secondly, during metaphase, when those chromosomes start to separate and pull towards the poles for anaphase to take place, they separate independently. So the paternal chromosome may go up and the maternal down, and for another characteristic, the maternal may go up and the paternal may come down. They separate independently every single time for karyokinesis. Okay, now we go into oogenesis. And oogenesis is gametogenesis in the female. So having a look here, we say, right, this is a cross-section through an ovary. All right, I just want to show you the whole diagram. This is a cross-section through the whole ovary. And being the whole ovary, here we're going to show you the different parts and the different processes that occur around the ovary. The biggest, most important thing that happens here is that when this egg is released, that is called ovulation. An ovule is released, and the ovule is your egg cell. All right, so we start here with our germinal epithelium. Okay, there's the germinal epithelium, and in that germinal epithelium we have the primordial follicles form. So the primordial follicle here, and it develops and gets larger. Then we have the primary oocyte, and around that primary oocyte we have the granulosa cells. This then develops into this structure, which is the primary oocyte, and it now has a primary follicle and a nucleus. All right? So this is the primary oocyte, and it develops, it grows into the structure here. All right, now, in fact, I'm going to just change my pen here. Okay, so from here, it's now going to develop into the secondary oocyte. The secondary oocyte has structures like, for example, a secondary follicle, because we had a primary follicle here. A secondary follicle, the shell around it, if we can call it that, is the theca. And then it has a fluid area inside, and then it has granulosa cells. And those granulosa cells there again are to protect and ensure that it is okay. From here, it will develop into our Graafian follicle. And the Graafian follicle, people, that is important. It's that Graafian follicle that swells and bursts. And when it bursts and releases that egg cell or the ovum, then that is ovulation. So the Graafian follicle is very, very important in the reproductive process of the female. So in here, we're going to have a nucleus. We're going to, but that nucleus at this point is haploid, remember? We have the secondary oocyte. We have a little stalk that starts to develop here. And the theca is this nice thick wall to protect this little egg cell. Okay? From there, the Graafian follicle moves and it gets nice and close to the wall and it bursts. When it bursts, what do we have? We have this secondary oocyte is released. And it's got its granulosa cells and they are there to protect it. But that secondary oocyte is released. Why? The Graafian follicle ruptured. It broke, it ruptured, it tore, releasing this egg cell. This is ovulation. And reproduction can only occur once ovulation has occurred, because ovulation is the release of that egg cell into the fallopian tubes. Remember, we did the female reproductive system and all the parts. It's the release of that egg cell into the fallopian tubes, because that's where reproduction occurs. All right? Then we have now another very important process here is this structure here that now the Graafian follicle releases the egg cell, and clearly everything closes up. And then it turns into the corpus luteum. And that corpus luteum's job is to release progesterone. And progesterone ensures that should pregnancy occur, that the baby, that little embryo, is able to grow, because it releases progesterone, which stops menstruation from occurring. All right, so very, very important. You've got ovulation, and the Graafian follicle then becomes or turns into, develops into the corpus luteum. And if pregnancy does not occur, in other words, there is no fertilization, so there is no pregnancy, then the corpus luteum will degenerate and menstruation will occur. All right, now let's have a look at the process. We have oogenesis. There are about 2 million egg cells are produced in the female, look at this people, before birth. This is why women undergo menopause. We get to a point in our lives when we no longer have egg cells. So we stop reproducing. We cannot reproduce anymore. And it's nature's way of, because as a woman gets, or as your physical body gets older, so the chances of carrying a baby become harder, okay, or it's not chances, it becomes harder to do. You get old, you get tired, and your body just can't cope with it. So what your body does is it says, well, we know this, and because having babies takes quite a lot out of your body, you know what we're going to do? You're going to reach an age where you stop producing eggs. So women have a finite number of eggs from birth. Males produce sperm when they start producing sperm. They produce sperm until the day they die. So if they die at 110, they are still producing truckloads of sperm. Whereas females stop producing, they have a finite number of egg cells. And as they get older, so they produce less and less egg cells till eventually the body says, look, I haven't got any more egg cells to produce. That's why generally infertility lies with the female. Because depending on what traumas there are at birth, those egg cells are destroyed. So a woman may, instead of having 2 million egg cells before birth, she may only end up with, I don't know, 150 once that little baby girl is born. All right, and so they're going to have egg cells for less time than someone who has a normal amount. So there are a whole bunch of things, but females have a finite number of egg cells. Males carry on producing sperm until the day they pass away. All right, so here we go. Now, they have 2 million egg cells before birth, but look at this. Only 4 to 450 egg cells develop into secondary oocytes, which means that if they've developed into secondary oocytes, they can then develop into egg cells, but only 400 to 450. All right, now, the oogonia undergo meiosis, which is the same as in the sperm, and result in the primary oocyte. The primary oocyte then undergoes prophase. And why have I written prophase? Prophase of meiosis 1 throughout childhood. So each primary oocyte just sits there. It sits there in prophase of meiosis 1 throughout childhood. This is before the girl has matured. All right? Then the oocyte is enclosed by a granulosa cell and forms a structure called the primordial follicle, and you must know this. All right? So the oocyte plus the granulosa cells forms the primordial follicle. And then the secondary oocyte develops and they are released from the ovary during ovulation. And that secondary oocyte is what will now be ready and develop into the egg cell. Okay, people, so girls, 2 million egg cells before birth, and by the time birth process and all the rest of it, there are only 400 to 450 egg cells that can develop into secondary oocytes. And then you've got those oogonia undergo meiosis, and we have the primary oocyte. And that primary oocyte sits there, and it waits in prophase 1 until ovulation is ready to take place. So the girl has matured, and she's now reached puberty. For every month that a female has a period and menstruates and starts releasing egg cells, because menstruation only occurs once egg cells are released, one ovary will release an egg cell 1 month, and another ovary releases an egg cell the next month. So the ovaries sort of take turns, and you have one egg cell this month, and from this side another egg cell, and another egg cell. Now go and calculate the number of years that a female is reproductively able to produce, and you'll know the number of egg cells that they have. Okay, a female is fertile from about the age of 12 to 50. Now, please don't turn around and say, yes, but so-and-so started her periods or started menstruating when she was 10, and so-and-so at 11. We're taking an average age, which is 12. All right, so from about the age of 12, some women start going through menopause at 45. Others will go through menopause at 50, and others go through menopause at 55, which means their bodies no longer have egg cells to produce. All right, so we've taken an average age of 12 to 50. One primordial follicle will develop into a mature follicle called the Graafian follicle. Now, the Graafian follicle, inside there, you're going to have your primary oocyte, which is now going to develop into an egg cell or an ovule. The primordial follicle becomes the primary follicle. So primordial, when you talk about primordial, it's like way back when, in the beginning. All right? So that then becomes, from a primordial follicle, it develops into a primary follicle. And you've got your granulosa cells around it to protect it. Then the cells of the stroma of the ovary form more layers called the theca. The theca, I'm going to put here, is like a protective shell. All right? And it contains blood vessels, which is important. The theca and the granulosa secrete the female sex hormones. So people, where do those female sex hormones come from? From the theca and the granulosa cells. All right? And the granulosa cells are all around that little primary oocyte. All right? So the granulosa cells and the theca produce or secrete the female sex hormones, which are progesterone and estrogen. The developing follicle moves to the surface. Now let's just go back here. Here we've got our developing follicle now, and you've got your Graafian follicle. It now moves right against the surface. That's what we're talking about now. It moves right against the surface, and it will then rupture. And when it ruptures, you've got your bulge develops. Then the oocyte undergoes meiosis 1. All right, now we've got our egg cell is ready to be released. And that bulge carries on and on and on until we have this bursting. And when it bursts or ruptures, I'm going to put it here, rupture. If something ruptures, think of a dam wall. If that dam wall ruptures, it just bursts. All right, so it ruptures, and that egg cell is released, and that is called ovulation. And this is exceptionally important in reproduction, because if the female does not have ovulation, no fertilization can occur. All right, the egg cell now enters the infundibulum containing the fimbriae. Now, fimbriae are, we did this when we did the female reproductive system. You've got at the edge of the fallopian tube are these little fimbriae, and they move and they move and they say, come to me, come to me. And the little egg cells now released by the ovary, and it's sitting there. And the fimbriae are doing this. And they almost draw the egg cell into the fallopian tube. But we've covered this already, so it's just to put it all into perspective. When the egg cell is released, the Graafian follicle changes into the corpus luteum. Now, people, this is very, very, very important. The Graafian follicle becomes the corpus luteum. And if, now this is important, if the egg cell is fertilized, okay, the corpus luteum secretes progesterone. If the egg cell is not fertilized, the corpus luteum will disintegrate. And it's that progesterone that keeps you pregnant. So remember, progesterone starts with a P. Progesterone keeps you pregnant. Estrogen, which is spelled O-E, oestrogen is for ovulation. So estrogen is for ovulation, progesterone is to keep that baby there if you are pregnant. All right. Now, here's question 2. It's taken from the National Senior Certificate Life Science Preparatory Examination for 2008. It says, now, look at what I'm going to do. You take your pen, in your hand when you read a question like this and you underline and make little notes and write words in for yourself. Okay? The basal body temperature, in other words, basal means basic. It's the basic body temperature, is the temperature taken in the morning when factors such as exercise and eating and drinking and emotional disturbances have no influence. So emotional disturbances would be like driving in the traffic in the mornings here in Gauteng especially, and I'm sure Cape Town too. So there are no emotional disturbances. You've just woken up and everything is wonderful. You haven't eaten, you haven't done exercise, you haven't had a cup of coffee. There we go. In women, the basal body temperature drops. Now people, this is very important. It drops before ovulation and then increases sharply a day later. So it drops before ovulation, then the body temperature will go up on the first day of ovulation, and as that occurs, then the temperature starts to increase. Okay, during menstruation, the temperature drops again to normal. So menstruation, which means fertilization has not occurred, we are now going to have the temperature dropping again. If the temperature stays high, then pregnancy is assumed or presumed. Okay, now Josephine and Patsy are two young, healthy women. Now look, they are two young, healthy women, and they recorded their basal body temperature for 28 days from the first day of menstruation. Okay, now the cycle starts on the first day of menstruation. So that would be day 1. Most women, and what is the norm, is a 28-day cycle. But it could be because of stress, it could be all kinds of physical reasons and hormonal reasons. Sometimes a woman could have a 23-day cycle or 25-day cycle or 26-day cycle. A 28-day cycle is the norm. It doesn't mean it is the only one. All right, so 28 days, and the first day is the first day of menstruation. The results are shown in the table. Study the table and answer the questions. Okay. Now, this is the kind of thing, people, that they can show at you in an exam. So have your wits about you. Here we go. Basal body temperature of Josephine and Patsy for 28 days. And here it is. So let's have a careful look here. They show you that's when menstruation occurred. All right? Now, I'm going to use my lines here. Okay? There's menstruation. And you can see she starts off at 36.5 and she goes, and 36.2 seems to be her normal body temperature, okay? And she's 36.3. So there's a 0.1 difference in Patsy and Josephine. However, have a look here. Remember, for ovulation, the temperature drops and then it rises sharply. So let's have a look where there is a drop in temperature. Look here. And we've got 36.1 and 36.1 from 36.2 being, and then look at this huge increase from 36.1 to 36.5 and the same says there. So what does it tell you? It tells you that here is where ovulation took place. Okay, and then we have ovulation temperatures are all nice and high, and then of course it carries on being high here, whereas here it drops. Now remember they said to you, just from the information you were given, they say that if you are pregnant, one of those indications, if you haven't gone for blood tests, is going to be that your body temperature, the basal body temperature, stays high. So look at this. What does it tell you here about Josephine? Her body temperature stays high. Patsy's comes down. This means that she is probably pregnant. And I mean, this would be as quick as 2 weeks after ovulation. So there is an indication for you. All right, now let's look at our questions. What have we done? We've looked at what they've given us, they've looked at the information they've given us, and we've taken what we know and we've applied it. So here we go. On which day of the menstruation cycle of the two women did ovulation occur? People, remember we said here, temperature drops. There it's dropped. And there's the drop. And then we said it increases sharply. So from 31 to 36 is a huge increase. It's 0.4 of a degree. So clearly this is when ovulation took place. So it was approximately day 13. All right. So then, what is the difference between the basal body temperature of Josephine and Patsy on day 28 of their menstruation cycle? So let's go back. On day 28, we have got, and I'm going to do this answer, the answer to that question here. On day 28, okay, so this is question 2.2. On day 28, what do we have? We have, and I've just forgotten this lady's name, so let's just quickly go up, Josephine. So we're going to say Joe's had 37.2. Patsy has 36.2. So we want to know the difference. So this would be 0. Although we don't have to write 0, but it's just for those that are mathematically not inclined. And 3 minus 3 is 0. So the difference is 1. 1 what? It's going to be 1 degree Celsius, people. 1 degree Celsius, and if you didn't know where I got that from, it tells you at the top here, 1 degree Celsius. So don't just write 1 degree. And this question was, I think, for 2 or 3 marks, but here they're going to give you 1 for your minusing and 1 for writing 1 degree Celsius. All right, we've done that question. Then next one, these measurements were made at the same time each day, in the same room, while each human was wearing the same type of dressing gown. Explain why these precautionary measures were necessary. Whenever you do any kind of test where you are comparing two variables, make sure that you keep everything else constant. So a thick woolen dressing gown versus a thin cotton dressing gown would have made a difference. The same time each day, so that you know that they have slept, they wake up, and you check their body temperature at that specific time. So it's the same conditions for everyone else. So what you're doing is you are reducing the variables that could impact on your results. And I'm not going to write that out. But that is something that applies to any question where you are going to be doing any kind of testing and getting data. In other words, collecting data and getting results. You're going to make sure that all other variables are kept constant. And in this case, you are looking at the same room temperature, because room temperature, if it's hot or cold, can make a difference. The time of the day can make a difference. As they said, you get stressed in the traffic and whatever. And the same kind of clothes that they're wearing. All right, so that's your precautionary method. What you are doing there is you're ensuring that all the variables are constant so that your results are not impacted, so that your results are accurate. All right, then 2.4. By interpreting the data in the table, which of the two ladies is pregnant? Well, we've already established that. The data in the beginning of the question told us that one of the indicators of pregnancy is that the body temperature does not come down to normal, it stays slightly elevated. Give a reason for your answer. Well, that was easy. We said that it was Josephine. She is, we think, pregnant. It's not inconclusive, but the chances are. And why? Her temperature remained high. Well, it's high. You can put in inverted commas. It was 1 degree Celsius higher than normal. Okay, and that would have given you 2 marks. All right, explain how basal body temperature, I'm just going to make this a bit higher. Explain how basal body temperature could be used as a method of contraception. Well, it's called the rhythm method. And it's a method that is applauded by the Catholic Church because they don't believe in using contraception, which is, I don't know, as you know, we can argue that, and I have no intention of arguing religious issues with you. However, it's called the rhythm method. And you use body temperature to determine when you are fertile as a woman. And clearly you are fertile when you are ovulating, people. So that would be when the body temperature is high. The indication would be if the body temperature drops, then you know, oops, we're now heading towards ovulation. Remember that your male sperm survive for approximately 24 to 36 hours. The female sperm, in other words, the egg sperm, survive for longer. They survive up to 48 hours. So when that body temperature drops, you abstain from intercourse. And that would mean that you would then be safe until your body temperature dropped to normal. That means ovulation is over. If, however, you wanted to conceive a baby, then you would know, as soon as that body temperature drops, now we must ensure that sexual intercourse takes place while ovulation is happening. So if you wanted to use it as a form of contraception, it would be called the rhythm method. So I've explained that to you. I've just written rhythm method here, but you have the decrease 1 day before, and then you are going to have an increase during ovulation. All right, and that's what would give you your 2 marks. Explain one disadvantage of using the basal body temperature method as contraception. Well, it is actually only about 20%, there's a 20% room for error, mistake process. All right, so that's one disadvantage there. Another disadvantage is you may have various aspects in your life that could change or alter that basal body temperature. And you would have to be very strict about it. You would have to make sure that you use the same thermometer in the same place on the same day at the same time to check your body temperature. So any variation from that, and you could make a mistake. And that is why it is not very good. All right. I just wanted to show you something else just while we're going through the process. I don't want to do menopause. I do want to do menopause quickly before we finish off because we've run out of time. With menopause, look at it. Meno, and it tells you pause. So menopause represents the end of the fertile process in a female's life. It's when menstruation starts to stop. The body starts to slow down. And this is generally at about the age of 40. So menstruation cycles become irregular from about the age of 35, 40, depending on the number of eggs that person, that female started off with when they became mature. Remember, each woman is going to be slightly different, but generally about the age of 40. Now, this is a problem because sometimes there is a problem when meiosis occurs, and the chances of having a Down syndrome child increase geometrically as a woman passes the age of 35, especially by the age of 40. The follicles become less sensitive to FH, so the eggs are less likely to be released. Okay, symptoms of menopause, people, you will find that you've got aunts and grannies and moms that will be going through this hot flushes, night sweats, vaginal dryness, irritability, fatigue, softening of the bones. People, this is very important. This is why as females, from about the age of 18 to 20, you should be taking extra calcium supplements in your diet. Because as you start to go through menopause, our bodies are designed to release calcium. Why? Because we carry babies inside us. That's our body's design, is to carry a baby. So our bodies don't store calcium as easily and as well as a male does. And that is generally why male bones are stronger than female bones. All right, our bodies don't store calcium, so we need to take in extra calcium to make sure that our bone densities are fine. From the age of 40, a woman should be going for a mammogram once every 6 months to a year, depending on your family history of breast cancer. And we should be going for bone density tests from the age of 40. Every year, you go for a bone density test. But you need to start taking calcium supplements now because of this. You have a softening of the bones, and that results in this, osteoporosis. Osteo meaning bone, porosis, the bones become porous. The symptoms can be controlled, now this is menopause, with hormone replacement therapy, and we abbreviate it for you, HRT. And what happens there is estrogen is taken in pill form, or you get implants which they put in underneath the skin, which releases the hormone slowly. And then we can also take natural stuff, which probably isn't as effective as a tablet, but you have your naturotherapy, and we can then look at taking evening primrose oil. And evening primrose oil is good for all females anyway, because what it does is it helps you to regulate premenstrual depression and things like that. So it helps us to get our cycles nice and easy, and it helps our brains to metabolize and process everything. When we talk about PMS, or premenstrual depression, or premenstrual stress, it's a very real thing, because subconsciously your body knows that, oops, I didn't reproduce. And quite literally, on a very primary functioning of your brain, your role is to reproduce. So when you are not successful, and you now have a lack of progesterone and estrogen in your system, that is what now causes or creates the issue where you now have a lack of hormones. And that then causes a slight stressed situation in your brain. So it is very real. In some women it's worse than in others, but it does happen. And it's because there is a lack of estrogen and progesterone, and it is just before menstruation occurs. And that's what causes the menstruation, which is the breaking of the lining so that everything can carry on. All right, and the whole process, the uterus can be prepared again should fertilization occur the next time. All right, but more about that in our next session. There we'll go through the exact process in the female system. All right, so have a good one. Study hard. Remember, what you put in now, you will get out and reap the benefits for when you write your matric finals. Have a wonderful time. Have a good week. Until I see you again, bye-bye.